introduction. I'm humbled to be invited to this esteemed speaker series, and I would like to extend my gratitude to the entire um, meeting organization team, and specifically also to Dr. Vasantha Rupasinghe, who has reached out to me over a year ago initially to give the seminar at this series. So finally, here we are, and I'm really excited to talk to you about two main projects that we have been working on in the lab over the last few years. And I'll talk in my first part of my seminar about a specific kind of bioactive lipids, and in the second part of my seminar about dietary peptides derived from whey protein, with a common theme on obesity and metabolic syndrome. Without further ado, I would like to give you a bit of an overview of why obesity is of interest to my research program. Well, obesity, as you might know, is a epidemic. It is a major cause of morbidity and mortality in Canada and around the world. And just to give you a little perspective, these are data from about 10 years ago, but they still more or less hold true to today. So over the past 30 years, obesity has tripled for Canadian youth and doubled for Canadian adults. And in fact, more than 1 in 5 Canadian adults have obesity. Many more are living with overweight. And in fact, this has also trickled down to children, where 1 in 8 Canadian children now is classified as obese. And obesity is the accumulation of excess adipose tissue. It wouldn't be such a problem per se if it wasn't for comorbidities that it is associated with. And chronic obesity has been connected to dysfunction of virtually all organ systems, and is connected to cancers, osteoarthritis, non-alcoholic fatty liver disease, and many others. A particular interest through our research is the connection of obesity to metabolic disease, chiefly among those type 2 diabetes, as well as heart disease, cardiovascular disease, which includes coronary artery disease, hypertension, as well as cardiomyopathy, or the weakening of the heart muscle itself. And type 2 diabetes and cardiovascular disease are major reasons for excess morbidity and mortality in the obese population. Now, obesity is or the reasons for obesity are oftentimes multifactorial and include environmental factors, behavioral factors, as well as genetics, even epigenetics. And just to give an example for environmental factors, some of the causes include how the food is presented to us today. All foods once looked more or less like this. It's food was mostly unprocessed, similar to the fresh produce that you see here. And unfortunately, nowadays many foods look more like this, which includes nutrient, calorically dense foods, highly processed foods with relatively little nutritional value. And you might have heard of so-called food deserts, where there are areas around the globe where the access to fresh nutritional foods is compromised. Now, what happens with obesity? over many years on the cellular and tissue level is that adipose tissue, which is designed to store fat primarily in the form of triacylglycerols, is chronically overwhelmed with having to store more and more of lipids. And over time, adipose tissue becomes dysfunctional. There is inflammation, there is insulin resistance, of which I will explain in greater detail in a minute. But what that leads up to is that the dysfunction in adipose tissue and the inability to store lipids leads to a spillover of fatty acids to tissues that are not designed to handle a high lipid load, and this includes the skeletal muscle and the cardiac muscle. And that can give rise eventually to insulin resistance in those tissues, meaning that the insulin, the glucose lowering hormone insulin is not able to signal properly through the insulin receptor and downstream to promote glucose uptake into the tissues. And in addition, mitochondrial dysfunction is very well connected to insulin resistance and also excess lipid storage, and both together might culminate in heavily altered energy homeostasis, both at the tissue level and systemically. We also know that pro-inflammatory adipokines, these are cytokines that are released from adipose tissue, are very much contributing to this metabolic imbalance and inflammatory signaling in tissues such as muscle and heart. And this, derangement in the metabolic profile and chronic low-grade inflammation can then give rise to insulin resistance and type 2 diabetes, as well as closely connected to that is cardiovascular disease, including heart muscle weakening or cardiomyopathy. Now, my lab has been interested over the last couple of years in an adipokine that is called autotaxin, in short ATX, and it's an interesting adipokine that functions as an enzyme. It cleaves lysophosphatidylcholine to lysophosphatidic acid, which is the smallest lysophospholipid, and it's small but mighty. It packs a punch when it comes to stimulating signal transduction pathways that are fundamental for the cell, and it signals through 6 known G protein coupled receptors to influence a variety of intracellular processes, including what we also know it also influences energy metabolism. Now, what got us interested in autotaxin and the lysophosphatidic acid axis is a study that was led by our collaborator, Dr. Erin Kershaw from the University of Pittsburgh, where we showed that in non-diabetic women, middle-aged women, autotaxin levels in the bloodstream are related and independently associated with insulin resistance. And I'm showing here a graph that plots a insulin resistance index termed homeostatic model assessment of insulin resistance and autotaxin against autotaxin, and you see that there's a positive correlation. Now, we and others have subsequently shown a similar relationship in other population groups as well. In addition, a talented former PhD student from my lab, Dr. Kenneth D'Souza, has shown that in cultured adipocytes that are made insulin resistant, meaning that insulin is not able to properly signal through the receptor and intracellular pathway, including AKT, to stimulate glucose uptake in the cell, that in those cells, autotaxin release is or secretion is drastically increased, and on the contrary, the insulin sensitizing adipokine adiponectin is decreased. And that also led to a marked increase in the activity of autotaxin in the cell culture media. Now, what the question at the moment was is, how does the autotaxin lysophosphatidic acid signaling pathway influence tissue insulin function and energy metabolism? And so specifically the questions that we asked were, how does autotaxin and LPA influence insulin receptor signaling? In my talk, I specifically focus on the activation of AKT. Furthermore, we were wondering whether mitochondrial dysfunction could be induced by changes in LPA signaling, and as I mentioned, changes in mitochondrial function are very much connected to the development of insulin resistance and type 2 diabetes as well. Lastly, we were wondering whether this pathway influences the ability of the heart muscle cells to contract properly. And these research questions we asked with a specific focus on obesity and insulin resistance. Now, to answer these questions, we utilized the mouse model that has a partial deficiency of autotaxin at the whole body level. And we fed these mice either chow diet or a high-fat, high-sucrose diet for 20 weeks to induce obesity and insulin resistance. Similar to what you would see in humans with chronic obesity. And we determined their ability to respond to insulin systemically, and we also looked at the plasma autotaxin activity and body weight. Now, mice on a high-fat, high-sucrose diet, if you just focus on the white bars here, compared to the chow fat, mice fed on chow diet, have increased autotaxin levels. And those that have partial deficiency of autotaxin have about 50% less circulating autotaxin and reflected in its activity as well, and that remains low even after high-fat, high-sucrose feeding. Now, interestingly, mice that had less autotaxin also gained less body weight on the high-fat, high-sucrose diet. So this is a chow-fed animals here, and this curve is the curve of the wild-type animals that are fed high-fat, high-sucrose, and you can see that those with mice with partial ETX deficiency actually gain less body weight. And that also translated to improved insulin tolerance, which is assessed by injecting a bolus of insulin into the animals and following plasma glucose clearance over time. And in fact, while wild type mice on high-fat, high-sucrose diet had very much impaired plasma glucose clearance, this was markedly improved with partial autotaxin deficiency. So having understood that autotaxin deficiency actually has, or protects from systemic insulin resistance and excess weight gain, we asked whether insulin signaling was altered in a muscle. The reason why we asked this question is also because the skeletal muscle is the major site for glucose disposal. And so insulin signaling alterations in the skeletal muscle very much affect whole body glucose homeostasis. So we looked at insulin signaling. I'm focusing here on the phosphorylation of AKT, a critical node in the insulin signaling pathway. And We assess this by immunoblotting analysis after injecting animals with either saline or insulin. And what you can see is that in a high-fat sucrose fed mouse, insulin-stimulated AKT phosphorylation is slower compared to chow, and that this is preserved in high-fat sucrose fed autotaxin deficient animals, suggesting that partial autotaxin deficiency does indeed protect the muscle from impaired insulin signaling in this diet obesity model. Now, this also translated to somewhat preserved glucose transport in the muscle of high-fat sucrose fed mice with partial autotaxin deficiency, as was assessed using radio tracer analysis in isolated soleus muscle. Now, a former master's student in my lab, Karine Zorera, also showed that the same is true for the cardiac muscle. In fact, we again see that while insulin signaling is impaired at the level of AKT phosphorylation in wild-type animals that are fed high-fat high-sucrose diet, we see preserved AKT phosphorylation in those with partial autotaxin deficiency. And we also then asked whether changes in mitochondrial functioning could be connected to changes in the insulin signaling and energy homeostasis. And so we measured the respiration of mitochondria when offered different energy substrates, including acyl carnitines, using the oxygraph, which is an equipment that we utilize frequently in the lab to assess mitochondrial function and respiration. And what you can observe here, this is data for the soleus muscle, is that you do see a decrease in oxygen flow when mice are fed, wild-type mice are fed a high-fat high-sucrose diet, but in the high-fat high-sucrose fed Etiox het mice, we do see, in fact, that they have preserved respiration, suggesting that substrate oxidation and mitochondrial function are indeed preserved in those animals. And similar data we also observed for the cardiac muscle as well. Now, lastly, we were interested in whether cardiac functioning would be altered with partial autotaxin deficiency. So we subjected isolated ventricular cardiomyocytes to sarcomere shortening analysis. We basically stimulated the myocytes to contract and then determined the ability of the contractile units, the sarcomeres of the cardiomyocytes, to shorten. And a parameter that is very useful in assessing the cardiomyocyte shortening is called fractional shortening. This is the percent to which cardiomyocytes actually shorten or contract. And as you can see, the wild-type animals or cardiomyocytes isolated from wild-type animals on a high-fat sucrose diet had decreased shortening compared to their chow counterparts, and sarcomere shortening was in fact preserved in ATX deficient animals, suggesting that not only do we see preserved insulin function, we see protection from mitochondrial dysfunction, and we see protection from cardiomyocyte dysfunction when we eliminate autotaxin partially from these animals. And so our working hypothesis has been that during obesity and chronic excess lipid storage in adipocytes, there is an excess secretion of autotaxin, which leads to the conversion of lysophosphatidylcholine, often contained in lipoproteins, to lysophosphatidic acid, that then signals through lysophosphatidic acid receptors to skeletal muscle and the heart, and that the effect of that is that insulin function, mitochondrial metabolism, and the contractile function of the cardiomyocytes is impaired, in essentially initiating a vicious cycle where insulin resistance is made more severe, is further impaired, and that induces a feedback loop to the adipocytes to even secrete more autotaxin. Now, with that, I would like to switch gears a bit and talk the last few minutes about the second project that we have been working on more recently in the lab. And besides our interest in bioactive lipids, we're also very much interested in bioactive peptides that are derived from food sources. And the reason behind this is that some bioactive peptides from foods, including milk and whey, have been shown to have some potential in influencing energy metabolism and obesity, even in the setting of insulin resistance. And there's a growing interest in utilizing functional foods and dietary peptides for the prevention and treatment of metabolic dysfunction. And some of the data that that is out there from previous studies has provided good evidence that peptides derived from milk, specifically whey, are potentially influencing various aspects of energy homeostasis. However, the question that was still unanswered at that time, largely unanswered at that time, was what is the effect of bioactive whey peptides on energy metabolism in the setting of obesity in vivo. And so we set out in collaboration with Dr. Chibuke Udenigwe, who was formerly at the Dalhousie Faculty of Agriculture and is now at the University of Ottawa, to answer that question. So we fed mice through their drinking water, in fact, peptides derived from whey. We specifically hydrolyzed whey using pepsin and pancreatin, simulating a gastric digestion. And the resulting whey hydrolysate containing the peptides was then added to the drinking water of the mice. And in addition to that, we also added the unhydrolyzed whey protein isolate as control. And we fed the mice a high-fat diet for 13 weeks and assessed assess the body weight gain, their insulin tolerance, glucose homeostasis, and inflammatory profile. What we found is that whey peptide addition to the drinking water exacerbated body weight gain on a high-fat diet. As you can see in this dark blue curve, compared to high-fat mice that just consumed water and those that consumed the unhydrolyzed whey. And in addition, these animals also displayed impairments in their glucose homeostasis, meaning their glucose tolerance test showed that blood glucose was significantly above the blood glucose of high-fat mice that either consumed water only or water supplemented with the unhydrolyzed whey. And in addition to that, we also saw that the HOMA-IR, a marker of insulin resistance derived from fasting blood glucose and fasting insulin, was actually markedly increased in whey peptide consuming mice, and suggesting again that there is marked insulin resistance in those animals. And that was connected with enhanced systemic inflammation, as evidenced by elevated MCP1 levels, macrophage chemoattractant protein 1 levels. And also we found that even in the fecal matter, we saw an, evidence of increased inflammation, as is shown here by high, elevated fecal calprotectin levels in whey peptide consuming mice. Lastly, we also examined the adipose tissue, which I stated earlier is somehow or is definitely or inflammation in the adipose tissue is one of the prime driving factors behind obesity-induced insulin resistance and comorbidities. And so we do see that adipose tissue MCP1 is dramatically increased in mice-fed whey peptides. And this is also accompanied by increased levels of the macrophage marker F480, suggesting macrophage infiltration. And as expected, the heightened inflammation in adipose tissue is connected to decreased levels of transcription factor PPA gamma, which is known to be suppressed by inflammation and is a key transcription factor regulating lipid homeostasis in adipocytes and other cells. Now, to make a long story short, we did look at liver and skeletal muscle as well, but due to time constraints, I'm not presenting the data today. This summary slide shows you the key findings from this in vivo study, where we looked at the influence of whey peptides on obesity-induced metabolic dysfunction and inflammation. And basically what we see is that whey peptides, at least in our hands, do lead to increased body weight gain. They do enhance or exacerbate glucose intolerance, hyperglycemia. There is evidence of systemic inflammation and inflammation in the level of adipose tissue, as well as other organs. And in addition to that, data that I'm not presenting today, but I would like to mention that we also found that whey peptides increased steatosis or lipid accumulation in the liver and skeletal muscle and contributed to mitochondrial dysfunction. So in a nutshell, our data suggests that whey peptides might not be a suitable nutritional source of dietary peptides for the prevention and treatment of obesity and metabolic disease, but more research is necessary to assess exactly how these peptides influence glucose homeostasis and mitochondrial function, as well as lipid metabolism. Now, lastly, I would like to acknowledge the many former and current members of my laboratory who have contributed directly or indirectly to these projects. And as well, I would like to extend a special thanks to Dr. Purnil Kunil, who very much operates the lab jointly with mine, and is involved in many of the research projects from my lab. I would like to also thank Dr. Udenigwe for providing us the peptides and for his continued collaboration with our lab. I would like to thank Dr. Andrew Morris for his collaboration on the lysophosphatidic acid autotaxin work, Dr. Erin Kershaw for her collaboration on the clinical aspects of the autotaxin LPA study, Dr. Vasilis Aidinis for the provision of the of the autotaxin of the mice with autotaxin deletion, as well as Dr. Daniel Kane for all his help with mitochondrial analysis, and the IMPART team for their support. And with that, I would also like to thank the many funding agencies who have provided support for our research program and has made all this research possible.
Wonderful.
I do have 2 or 3 slides about the Just Dalhousie Medicine New Brunswick. I don't know whether it was in the realm of the timeline, or are we already past?
We are a bit past, but please, if you want to, please share.
Maybe just a minute. So for those who don't know, my lab is located at Dalhousie Medicine, New Brunswick, at the UNB St. John campus. And we established the first center in New Brunswick for discovery and translational research in cardiovascular and metabolic sciences. We welcome applications from interested students who would like to join our program. If you would like to get in touch with me, you do have my email address in the first slide of my talk. Thank you.
How rich that was. Thank you so much. So we welcome conversation now. Participants in the call are welcome to unmute their mic, as well as to write the question in the chat box, whichever you prefer. But please do feel free to raise your questions. And as we are anticipating and looking for questions from our colleagues,